Hi there, welcome to the Natural Products Atlas. My name's Roger Linnington, and I'm going to be walking you through the online curator, which the volunteer curation team can use in order to add new data to the atlas itself. So in order to do this, the first thing we need to do is go to the curation website, which you can find at npatlas-curate.chem.sfu.ca, which you should have been sent in the email that we sent you about curation. And this takes you to the landing page for the curation website, and if you go to the login tab on the top right here, it will give you the login page. Enter my login details, and that will get you into the center. So this dashboard landing page inside the curator shows you which data sets have been assigned to you. And the reason that we use a login system is that it allows us to track which data have been assigned to which people, and to provide provenance for the data which we insert into the atlas. But it also allows you to see just the things which are assigned to you to curate and to see how far through that curation process you have progressed. So on the datasets page here, you can see that I've only been assigned a single dataset. And if we click the See All Articles link here, you can enter into the dataset and you can see row by row which the articles are which are assigned to you, and then what's happened to them, whether or not they have been completed. You can see here on the right, as well as some details about the name of the journal and the article and things like that. If you click Dashboard again here in the top right, it'll take you back to that main landing page. And then if you want to start curation, you would click up here on the left. It will either say Start Dataset if you haven't done any work yet, or if you've already done some curation, it will say Resume Dataset and allow you to pick up where you left off. So I'm going to resume this data set. And when I do that, you can see that we get a pop-up window. These are instructions which pertain to that particular data set. So the inspiration for which compounds to include comes from a variety of different sources. And so there may be subtle differences or some variations between the different data sets. And so it's a good idea just to read the instructions and see what they say and make sure that you know what you're supposed to be looking out for and what you're supposed to be curating here. So here you can see this is a sandbox data set, which allows us to sort of mess about during the curation demonstration without risking the data itself. So I'm going to say OK, and it will then bring up the page for the next article in the series which needs to be curated. So this curation process is an article by article process, and so what we're asking curators to do is to look at the information about each article to determine whether this article is really about new natural products or not, and if it is, to make sure that all the new natural products that are reported in this article are listed in the compound section. So for us to do curation here, the first thing I would do is I would look at the title. So fomastin I, 13-epi, fomastin I, and fomastin J, 3 novel diterpenes from a marine-derived fungus. So it's important that it's from a microbial source. So we do not want papers from invertebrates or plants. And then it's important that it reports new natural products. And so you can see in the abstract here it says 3 novel diterpenes were isolated. So that seems very good. In the bottom here, there's a flag for the number of compounds expected from this paper. So these are flags that have been added by other curators previously. And the reason they're in your data set is because the number of compounds we have doesn't match up with the number that are reported. And there are 3 reasons for this. It could be that the number of compounds reported is erroneous and that we actually have all the right compounds, in which case you can just change this number. But in this case, we know that the number is correct. It could be that we don't have all the compounds yet, which is true in this case. Or it could be that 3 compounds are listed in the abstract, but only 2 of them are new, for example, in which case whoever originally annotated the number of compounds did not annotate that correctly. So in the end, in order to proceed, the number of compounds in this box needs to match the number from the curated compounds list, which is in the compound section here on the right. So if I click on this drop-down tab, I can see a list of all the compounds. At the moment, there aren't many in this page, so this seems redundant, but if there are articles that have many compounds, sometimes it's useful to see them in this list format. And then underneath, you can see tabbed region, which shows the compounds one by one. So at the moment, this tab is slightly darker, so it's showing fomactin I. And if we want to look at the structure from fomactin J, we can click on this. They are a little bit different to one another. Yes, you can see here. So this double bond in fomactin I is then a different functional group in fomactin J. So that seems reasonable. These tabs here are in green because these are compounds we already have in the atlas, which we have already curated. So the expectation is that those should be of good quality. Sometimes you also see tabs that are in yellow, and there we've used other computational tools to look at the abstract wording and identify other compounds which we think should be new, and then to go and try and find those compounds from public databases like PubMed and other places. In this case, the tool did not recognize any additional compounds, and so we have to find that compound by hand. But things that are in yellow are definitely things you should assess, because they may be correct or they may be incorrect, so you have to look at those quite carefully. So in order to find the remaining compounds, we only have two choices really. One is that we can try and find the structure of the missing compounds from a public database like PubChem, and the other is that we can draw that missing structure in some drawing program and then paste the data in as a new compound. But before we do either of those things, we really want the article itself. So here you can see the DOI, and next to the DOI is a little outlink here. So if I click on this outlink, it should take us straight to the page for that article, and here it is. So that looks great. So you can go directly to the article page, which is very helpful, and then if I click download PDF, we should then download the PDF, which we do. So I'm going to open the PDF here from my download section. There we go. So here's formactin I, 13-epi formactin I, and formactin J. So the compound we're missing here is 13-epi formactin I, and here in the first page you can see that the difference is only in the relationship here on the six-membered ring. So if I go back to the Curation tab, the easiest way for me to get the structure in this case is to take the smiles, highlight the smiles here, and copy it, and then go to ChemDraw, let that load, and now here in ChemDraw, go Edit, Paste Special, Smiles, and it gives you the structure. And we know that 13-epi is only epimeric at this one center. So I can just change that position, select this compound again, edit, copy as smiles, and now I can go back to the curator and I can say add compound. And that will produce now an empty box into which I can paste the smiles. I can also go to the title, copy, and paste the compound name. And provided that the source organism is the same as the other ones that are in this paper, this will already be filled in. So in our case, the fungal identification was not done by the original authors, and so we don't know the identity of that fungus, and that's fine. So now, if we go back to formactin I, and then we look at 13-epi formactin I, unfortunately they're displayed flipped to one another, but this is the center that's changed, and so that looks to be fine. So now the number of curated compounds matches the number of compounds we expect. Those compounds are correct. Everything else in the page looks accurate. So now if we scroll down, we can say submit data. So when we do that, we now get the next in the series. And this is an article from the Journal of Antibiotics from 1991. So in this case, it's saying that parahequamide was isolated from penicillium along with 3 novel analogs, 2, 3, and 4. So in this case, the original compound is not new in this article, but 3 novel analogs, 2, 3, and 4 do need to be added. And it looks like we've got one of them, and it has a name. So we'll have to go and check that in a minute. You can also see that our algorithm has recognized parahequamide as a natural product name in the abstract, and so it's given it to you as a candidate. But in this case, that candidate is not correct because that candidate is not new in this paper. So I'm going to use the garbage can tool here just to delete parahequamide, and that should then load with just a single example, and the curated compounds number has gone down to 1 now. So we can go again to the DOI link here. So I'm going to go link out now to the Journal of Antibiotics. Let's see what we see. Okay, so here we are in the Journal of Antibiotics page. We can hit download PDF. We should get the PDF from them quite quickly. There it is. And now we can open that PDF. And here are compounds 2, 3, and 4. So I'm going to zoom in on this PDF a bit so we can see what we're doing. So here they are. So they vary a little bit in their functional groups here. And you can see that they've got code numbers which have been assigned by the authors. And so these are the code numbers we need to add as the names here. So again, there are two choices for how to find these compounds. One is to take them and modify them. So in the case of these compounds, they don't vary very much from one another. So it's probably easiest to take the existing example and just modify it slightly in ChemDraw. That's probably the fastest way to do it. Let's just look back very quickly at which compound we already have. So we have VM54159, and if I look back at the paper, 54159. So another one that we don't yet have is VM54158. So the other mechanism for getting compounds is to go to PubChem. So I'm going to do that here, PubChem. And here I'm going to say VM54158, search, and let's see what it says. Okay, so we were unsuccessful in finding that compound in PubChem. There are other databases that you can look in. You can look in ChemSpider, you can look in Chembl, but if PubChem is not successful, often it is quicker to redraw than it is to work at looking for them in all the other public databases. So in this case, we would then add a compound for 54158 and add another compound for the other compound which is missing. That would bring the total up to 3. It would then match the number of new compounds, and we would be able to go down and submit. Let's say, for the sake of argument, that there's some problem with the structure, and either the structure is very hard to interpret, or it's very difficult to get the structure, or drawing the structure is going to take an enormous amount of time. If you try and submit in order to move on, then it won't let you do that. So if I try and submit here now, it will say error in the number of compounds field. Reported number of compounds does not match the number of created compounds. So you can't move on in that way. So if you want to move past a particular paper, there are two ways in which you can do that. So the first way is that there is a checkbox here called needs work. So if you have looked at the paper and you know that it's going to take a lot of work for somebody to figure out how to sort this paper out and get the right structures in there, then it's okay to check the needs work box. And now if you hit submit, it will submit, and we can tell internally that this is a paper which we're going to need to give to a specialist who's going to need to spend some time on it. If alternatively, you know that you can do this paper, but you're in a hurry to do something else, or you haven't got time to deal with it right now, you can hit skip. And if you hit skip, it will stay live as one of the papers that you have not yet dealt with, and it will give it back to you again next time you come back to the data set. So for now, I'm going to skip this one, just so we can look at another example. Okay, so here's another example. This article is from the journal Bioscience and Biotechnology and Biochemistry. There are 4 compounds listed. We have 3 of them here, so it's clear which one we are missing. We have a DOI. Everything looks good. So this would be a fairly straightforward case where it would be easy to go and look for the outstanding compound and find its structure and add it. And so the curation really goes along in that way, looking for compounds, finding them, adding them to the data set, and then once the data set is complete, then we will take the completed data set from you, run it through our internal quality control checks to make sure that things like the journal name is consistent with the journal names we already have in the database and things like that, some basic data handling, and then those results will be inserted straight into the atlas. Perhaps I'll do one more example here, and we can follow this through, and then we'll finish off for today. So if I follow the DOI link again here, it should take us straight to the article. Here it is. It may be that your library doesn't have access to all these articles. We have found in general this works better on campus than off campus, that the links tend to work a bit better. But there may be some that you just can't do. In this case, this is one that we do have. I can look at so here are the structures. I'm going to zoom in again a little bit so we can see what's going on here. And in this case, you can see that A and C are very similar to one another. In fact, they're very similar. So the easiest way to obtain the structure then is to go back to our Curation tab. We're on the tab for A. I'm going to copy this smiles. I'm going to go to ChemDraw. I'm going to delete the old structure. And then I'm going to use the shortcut key to paste smiles. Oops, I think I messed that up. Let's try again. Let's copy the smiles here. Go back to ChemDraw. There we go. Paste that in. And then we know from the original paper, which is here, that in the case of C, all 4 of these groups are methyl thioethers. So in that case, we need to just add that functional group here, like so. Select the whole thing, edit, copy as smiles. There is a shortcut key for copying smiles as well. It won't work if you just use Control Copy, but if you use the special shortcut key, then it works fine. And then back here, you can say Add Compound. Here it is. There's the smiles that goes with it. Compound name. I'm just going to copy from the title. In general, it's better to copy these things than it is to retype them because you make fewer mistakes. So there we are. That's the compound name sorted out. And then that should show up here. So now the number of curated compounds is 4. The number of compounds it's expecting is 4. Everything looks good. We're in a position to submit those data. And that's it. So that's how the curation gets done. If you've got any questions, you can contact us through the email system that we're already using to communicate with you, support at npatlas.org or volunteer at npatlas.org. And so yeah, we wish you luck. We hope that it goes smoothly, and certainly very appreciative of your effort to help the community with this new project. So let us know if you run into any difficulties or if you've got questions. You can also read the frequently asked questions PDF that we've generated, which gives you a few more details about what you should include and what you shouldn't, and sort of how to process the data. Hopefully that will be very straightforward. So yes, thanks again for all your effort.